Hello, and welcome to this asynchronous AICHE talk on viral replication and cytokine responses. I'm Jordan Weaver, a third-year PhD student in the Shoemaker Immunosystems Lab at the University of Pittsburgh. Let's begin with the motivations behind this work. Respiratory infections are a constant threat to public health, with deadly infections often characterized by cytokine storms, that is, an overly aggressive innate immune response that can result in severe, unnecessary lung tissue inflammation. Influenza causes an annual 49 million infections and 79,000 deaths in the United States alone. The disease also presents a significant pediatric burden, causing 374,000 annual hospitalizations of children under 1 year old globally. The new severe acute respiratory syndrome of the novel coronavirus, SARS-CoV-2, has emerged, resulting in over 220,000 deaths in the United States to date. Both influenza and SARS-CoV-2 are RNA viruses, and the data suggests that cytokine storms are a common feature of both viruses during severe infections. Immune responses can help or hinder an organism's ability to overcome an infection, and excessively inflammatory responses, like cytokine storms, can cause greater tissue damage, higher mortality, and slow recovery. Vaccination is effective for protecting public health against seasonal influenza. However, when new strains unexpectedly emerge, such as the 2009 novel pandemic H1N1 virus or the 2019 SARS-CoV-2 virus, new treatment strategies that can be implemented rapidly, and preferably independently of the specific virus, are needed. Immunomodulatory treatments that aim to reduce inflammation while still managing virus growth are a promising approach to protecting against emergent disease, but several fundamental questions on how these unnecessarily aggressive immune responses emerge remain unknown. Mathematical modeling can help quantify the kinetics of the interactions that define the immune system, revealing the interactions that are most responsible for the unnecessary aggressive responses and potential targets to interfere with immunity and ensure healthy viral clearance. Current models of innate immune responses to RNA virus infection are frequently numerically unstable. The size of these models, referring to the number of parameters and ODEs that they consist of, prohibits the analytical approach of steady-state analysis. Additionally, many existing models lack major intracellular components or lack important biological interactions, limiting their applicability to understanding how severe inflammation emerges. A few of these missing interactions that we sought to include were interferon-stimulated gene or ISG effects on inducing a virally resistant state, viral self-amplification, and how sensor proteins and paracrine signaling affect the dynamics of the immune response. Extensive molecular pathway maps exist, but they currently lack the mathematical description to support simulating the immune response. Some models of the intracellular innate immune response have incorporated RIG-I and toll-like receptor sensor family activity, but consider their effects to be constant, independent of the viral load. Finally, most existing models are trained to a single viral strain, which greatly limits their applications. I'll gloss over the construction of our model and just give you the final structure. Parts of this model may seem familiar. It is essentially a spiritual successor to the standard model of viral replication, which now incorporates cytokine signaling and host innate immune responses. I'm not going to go into great detail here. You can dig into our MDPI Processes Journal article if you really want the nitty-gritty details of the model. For now, I'd like to highlight a few important aspects. First, the two families of virus-activated sensor proteins, RIG-I and the toll-like receptor family, are incorporated here distinctly, highlighted in red. Second, cells are capable of upregulating Type I interferon production via paracrine signaling and the JAK-STAT pathway, highlighted here in yellow. Finally, the cell's attempts to fight off invading virus via interferon-stimulated genes is highlighted in blue. Note that only 4 parameters of the model are specific to any given virus: K61, K71, K72, and K73. This means that the model can ideally be trained to a new virus with very little data or computational cost. We'll get into why a bit later, but this model was designed with a minimum number of ODEs and parameters to keep computational overhead as low as possible while still capturing the full dynamics of cell behavior, cytokine release, and viral replication. The model consists of 7 ODEs and 16 parameters which couldn't be sourced from literature. We used a parallel tempering Markov chain Monte Carlo, or MCMC fitting algorithm to train this model to an influenza infection of human bronchial epithelial cells in vitro. Data are represented by filled blue circles. Solid black lines give the best fit of 1 million MCMC iterations, while the gray area is plus or minus 1 standard deviation among the best 1,000 parameter sets, essentially showing the confidence interval of our parameterizations. Of course, nearly any model can be fit to data if you hit it with fitting algorithms hard enough. The true test of a model is its capability of predicting outcomes in a system it's never seen before. Our validation study was an infection just like our training data, except that the influenza virus was incapable of producing non-structural protein 1, which changes the way sensor proteins are detecting the virus. Dotted lines here are the original model training from last slide. Blue circles are data from the NS1 knockout strain. Solid lines and the gray area here are entirely predicted, not trained directly to this dataset. In the validation, I mentioned non-structural protein 1 changing how cells detect the virus. We can gain insight from this model on the behavior of RIG-I and that exact change. It's vital to the detection of the invader and the initiation of the innate immune response. The stronger the activity of RIG-I, typically, the better the outcome for the host organism. This sensor protein is frequently targeted by viruses, for example the NS1 protein from influenza and NSP1 from the SARS family. The severity of influenza strains is often correlated with the efficacy of this antagonism, and that trend seems to hold for SARS. Here we can see that RIG-I has adapted to have a robust action, where even a 75% antagonism of the sensor from the moment a virus enters the cell still results in significantly better outcomes for the host organism. There's a second part of the puzzle, paracrine signaling. Infected cells secrete Type I interferons, which are detected by the JAK-STAT pathway in neighboring cells. This detection leads to the production of even more interferons, propagating the signal and warning cells in advance of the virus. If this signal is strong enough, interferon-stimulated genes, or ISGs, can halt the virus's progression entirely. These graphs are the case of high multiplicity of infection, or MLI, that is, every cell is initially infected. In panel A on the left, an NS1 knockout strain of influenza is allowing RIG-I to act to its full potential. You can see that sensor proteins are responsible for a lot of the interferon production early on, and that paracrine signaling takes over as we approach about 24 hours post-infection. In panel B on the right, the wild-type influenza has almost completely shut down sensor proteins. However, enough activity remains that some interferons are successfully produced, and the paracrine signaling pathway takes over, accounting for about 98% of interferon production. These effects will vary in lower multiplicity of infection simulations, and are amplified in the presence of immune cells, some of which can further amplify the signal, but it is illustrative to see paracrine signaling's contributions to cytokine responses. The reason that we kept computational overhead low for this model was in anticipation of implementing an agent-based model. This spatio-temporal environment has a slightly modified version of the ODE model, with per-cell integrity and infection state. This isn't in the processes article, but do look for a preprint with this development soon. Some highlights of the agent-based model are the ability to predict plaque growth in vitro, lesion growth in vivo, seen on the right, and start to introduce immune cells' spatially dependent actions. Thank you for your attention. While I would have liked to include a picture of the current Shoemaker Immunosystems lab, the pandemic has made that impossible. Instead, I'd like to thank the University of Pittsburgh, Dr. Jason Shoemaker, the members of my lab, and Dr. James Glazier and his lab of Indiana University. I'll be hosting a live Q&A session from 8 to 9 a.m. Pacific Standard Time on Thursday, November 19th. The Zoom information is provided here and also available through the live session section of my AICHE iPoster. These slides will also be available through my iPoster.